Good afternoon, everyone, and a very warm welcome to today's APPS seminar series, second session for 2026. My name is Shreya Patel from Northern Territory Department of Agriculture and Fishery. I am the Regional Counselor for Northern Territory, and with a great pleasure, I will be the moderator for today's session. Before we start, I just want to mention a few housekeeping items to ensure everything runs smoothly. So please keep your microphone muted and camera off during the presentation, unless you are presenting or while you ask the questions. Feel free to drop your questions in the chat box during the presentation, and we will have 5 minutes dedicated Q&A time, and we will address it during that time. And just a heads up that today's session is being recorded and will be available on YouTube for later use. Before starting, I would like to acknowledge the traditional owners of this land on which we live and work, and I pay my respect to elders past, present, and emerging. We have a fantastic two excellent presentations today. And with a great pleasure, I would like to introduce our two speakers. So first speaker is Dr. Melanie Ford, who is a nematologist at Northern Territory Department of Agriculture and Fisheries here in Darwin. She is currently working on the NT component of the Hot Innovation Vegetable Funded Project, Guava Root Knot Nematode Identification and Management. Today in this presentation, she will outline the species of root knot nematodes identified across the territory during this project, along with the susceptible crops, weeds, and ornamental plants. It will give us insight into an understanding of distribution of these root knot nematodes and the susceptibility of crops grown in Northern Territory. Now, I would like to introduce our second speaker for today is Dr. Sonu Yadav, who is a principal molecular scientist at Northern Territory Department of Agriculture and Fishery here in Darwin again. Sonu leads and manages the diagnostics and research in molecular lab in this department, and she has supported next generation sequencing and metabarcoding research projects in past few years. And she is going to share one of these study today with us. She will be taking us through her metabarcoding research project to characterize fungal composition across 4 climate zones in Northern Territory over multiple seasons. This work provides valuable insights into soil fungal diversity and relative abundance across diverse ecosystems, establishing baseline data to support biosecurity surveillance and environmental monitoring. So without further delay, let's get started. And I would like to invite our first speaker for today, Dr. Melanie Ford. Whenever you are ready, Mel.
Thank you, Shreya. So Shreya just covered, I'm going to be discussing root knot nematodes in the Northern Territory today. And I'm part of the Biosecurity and Animal Welfare team at the Department of Agriculture and Fisheries. Now, before we get started on root knot nematodes, I thought I would give you a little bit of a refresher on nematodes in general. They're one of the subjects that we cover, but not as often. So nematodes are microscopic roundworms, or they're mostly microscopic. There's some that are a little bit bigger. There's an estimated 30,000 different species, if not more, on Earth, and many live in the soil and are beneficial. As you can see from the electron micrographs on this slide, there's a lot of different trophic groups. So we have bacterial feeders, fungal feeders, omnivores, plant feeders, and predators. And the beauty of this is if you start looking at the diversity within your soil, you can actually use that as a metric for soil health. It's pretty interesting. Now, on the plant feeder side, we often call them plant parasites. There's about 4,000 different species, and 10 are really economically important worldwide because of the damage they cause to our crops. So one of those 10 are root knot nematodes. Now, all root knot nematodes are in the genus Melatogyne, and they are a sedentary plant parasite, which means they have a motile juvenile stage where they're in a worm-like shape, and they swim through the soil to navigate to a plant root. Once they get there, they actually go inside the plant roots and find a feeding site. So at their feeding site, they start to inject some chemicals into the plant that causes it to reprogram the cells around it. The cells will become multinucleate and absolutely gigantic, hence the very technical term giant cell. And what happens then is the plant begins to feed the nematode, and the females will spend the rest of their life cycle inside that plant root. Now, as these giant cells develop and the nematode grows bigger and changes shape, it starts to form what's called a gall. And that's where they get their name, root knot nematode. It forms this big lumpy ball around the nematode itself, and the plant roots look really lumpy. That becomes a problem with our crops just because the nematodes are taking all of the nutrients out of the plant. So symptoms often look like stunting, wilting, and nutritional deficiencies. It's incredibly hard to recognize nematodes just from the surface. And one of the other fascinating things they do is their reproduction. So the males generally aren't involved in reproduction, and most of the time it's done by the females through parthenogenesis. So each female is able to clone herself in all of her eggs, and she can produce up to 500 different eggs at a time. Now, within the Northern Territory, we know that root knot nematodes have been a problem for quite a while. We have some historical samples that have been collected all the way back to the 1950s, and in our collection, our samples basically range from the 50s straight into present. Now, we know that in the past, most of the root knot nematodes identified in the territory have been two different species, and that's Melatogyne javanica and incognita. Now, it's incredibly difficult to ID them via morphology because they look so similar. You have to have a really, really good microscope. And what you do is you pop the females out of the root underneath the microscope and then micro dissect those females. And you're looking for this one particular thing called a perennial pattern that's kind of on the back of the nematode. And there is a bit of a shape in the perennial pattern that acts as almost like a fingerprint for identification. Now, it's incredibly hard to do that, and so some of the morphology and identification of nematodes has been missed in the past, and they have been misidentified, including one we're guessing has been misidentified, and that's Melatogyne enterolobii, which is the guava root knot nematode. So that's a newish nematode to Australia. It was first detected in 2022 in the top end of the Northern Territory. And we think it's been around for a little bit, at least 5 years, because after the 2022 detection, we started going back through some of our DNA samples, and we realized it was detected in 2021. We just had misidentified it. So because of all of these issues with identifying these things and the difficulty with it, Hort Innovation, AusVeg, and multiple state and territory governments have gotten together for a big project. And this project has been the guava root knot nematode identification and management. Since it was first found in the Northern Territory, our portion of the project contains 3 different kind of tiers. So the first is improving the diagnostics of how we're going to ID these different types of nematodes, especially the root knot. So one of those portions has been really delving into the molecular biology and trying to come up with different ways to identify it and to boost our capacity to do that. The next is surveillance, and that's conducting surveys across the Northern Territory to understand the distribution of root knot nematodes, which species are where, and how they're impacting crops. And finally, we also want to come up with different management strategies. And we know that rotating your crop with a commonly used cover crop is often a good strategy if you're using a nematode-resistant cover crop. However, we're not sure if a lot of the commonly used cover crops in the territory are resistant. So a portion of the project has been challenging our cover crops in the territory under glasshouse conditions to inoculum and testing for that. So describe the different portions of the project. The first, as I mentioned, has been the diagnostics. And what I've really been focusing on is optimizing a multiplex assay. So this is a PCR-based assay, and it's been playing around with different primers and conditions to kind of get it right for us in the territory. Up here, we have 4 different species of root knot nematodes that are really common. So what I wanted to do was be able to test for all 4 species at the same time to kind of simplify the assay in ways where I could just test a bunch of different samples to see which species I have. But the other important part is being able to use this assay to determine if I have mixed infestations of multiple species on the same plant. And so far, we've been able to kind of optimize things, and we've come up with an assay. From the gel on the right side of the screen, you can see it's a little bit complicated to interpret, but that's kind of the gist of it. Now, once we had some diagnostics techniques that were pretty solid, I started doing surveillance. So for the past 7 months, I've been traveling around the Northern Territory, and in total, I've collected 260 root samples from the major growing regions across Northern Territory. And that includes the top end, Douglas Daly, Catherine, and Central regions. Now, I've been preferring to collect root samples over soil, as one of the aims was to determine the susceptibility of different crops in the Northern Territory to guava root knot nematode or any root knot nematodes under territory environmental conditions. As we all know, our environment up here is a little bit different to the rest of the country, so we needed to see what was happening for us. Within this, you can see on the graph that I've kind of looked at the different percent of detections of each species. So from this graph, you can see that roughly 30% of the detections are the guava root knot nematode, the Melanogyne enterolobii. Not many are in Arenaria. That one's predominantly in bananas, and up here at least. Melanogyne incognita is also common. And what was interesting to me is that Javanica actually seems to be the most important species. While only 20.6% of detections were Melanogyne javanica on its own, it's also the nematode that's always found when there's co-infestation, so multiple species infesting the same root. And in total, that means that 30% of all detections in the territory include Melanogyne javanica. I should also point out that the top bar there for the Melanogyne spa, most of those are ones I have been unable to identify as of yet, and it's mostly because I have a bit of a backlog of samples that I'm doing diagnostics on. So I do know that we have those samples in, I just haven't run the PCR assays on them yet. Now, to break down those different detections, I found that root knot nematodes have been present in 30 different crop species across the territory. And from the table that you can see on the right, that includes all 4 of the species that we have. I have been testing for 2 other species, which are hapla, Melanogyne hapla and Melanogyne phalanx, but I have not detected them in the territory to date. What's also interesting is that I've only found enterolobii, the guava root knot, in the top end and Catherine regions. So it seems to be at this point kind of within the northern portion of the territory. And what's interesting are where I'm finding it. So after all of the sampling, it's been pretty interesting to see that it seems to be always associated with guava trees. So it's either in the northern suburbs, or if it's on a commercial farm, it tends to be in the guava trees and any crops that are planted adjacent to the guava. I've also found it in some weeds and ornamentals. And the weeds are something that I have been slightly concerned with because a lot of times the weeds can act as hosts. So even once a crop is finished, if there's still weeds within a block, those can host those nematodes, and those populations can carry over to when the next crop is planted. I've also made a few different interesting observations. As I was going through some of the historical samples and some of the literature here in the territory, I started looking at, in the past, what's been recommended for management strategies. And one of the techniques for the central region of the Northern Territory, which encompasses Alice Springs and the Central Desert, was in the past, everyone was just told if you have a root knot nematode problem, turn off the irrigation and just let your crop die. And that'll get rid of your problem. It'll completely starve them out. And then in a year or so, you can go back, replant, and your problem will be solved. I found anecdotally, I'm not quite sure if that works. I was able to sample at an abandoned vineyard, and that was an interesting one. It was abandoned in 2021 due to COVID export issues, where they turned off the irrigation, they stopped all maintenance, and walked away from the property. So essentially, by the time I was sampling, the vines had been abandoned for over 4 years. They've been through 4 different summers in just outside of Alice Springs, with temperatures frequently exceeding 50 degrees. Now, as you would expect, a lot of those vines died, but about 1% were actually still living, which kind of blew my mind. So I decided to target those live vines and see if I could find any nematodes in them, which involved a lot of digging in very, very hard soil. But when I was able to extract roots, I was quite surprised to find they were heavily galled from nematodes, and I did extract healthy Melanogyne javanica from those roots of those vines, which really was a bit of a lesson for me to learn, because it means that turning off the irrigation isn't necessarily a good strategy. You have to make sure those plants are dead. If those vines are still persisting 4 years on, we still have populations of root knot nematodes within that soil. Which is why it's more important for us to start diving into other management tactics. So, as I mentioned before, planting cover crops and rotational crops are a really good strategy to break that cycle. If you plant something that is resistant to the nematodes, you basically can starve out the populations that are existing within the soil. So what I did in the glasshouse is I started looking at 6 different commonly used cover crops across the Northern Territory. Some of you may be familiar with these, things like pronto sorghum and stuff like that. Others might be a little bit more niche. Cavalcade is a common hay crop up here. We also have Rhodes grass, Milgar butterfly pea, sun hemp, and wind cassia. And I combined those with a positive control, which is tiny Tim tomato, and a negative control with the jumbo sorghum. So the goal was to inoculate with about 5,000 eggs per plant and incubate them. Now, nematodes, their reproductive cycle and their life cycle generally follows heat hours. So it's based on the soil temperature. So I incubated these for 14,000 heat hours, because at that point, we know that the eggs can go, or the nematode can go from one generation of eggs to the female reproducing the next generation. Once I grew out these inoculated plants, I harvested and extracted all of the eggs. I did egg counts as a metric for host susceptibility status and looked at the reproduction factor. And so that's just looking at the final population of eggs divided by the initial population that I inoculated, which was 5,000. And then looking at the data, because I had a negative control and some others that I thought might be resistant, those numbers are quite low, sitting around 0 really. And then ones that are susceptible have really, really high numbers, sitting around like up to 300,000. So I had to use a Poisson regression in a generalized linear mix. I can't even talk today, sorry. Basically, I used a Poisson regression to model the data. And really what that looked like is we were pretty successful in finding quite a few things that are resistant. So everything that's now down near the bottom, sitting around 0, especially for a reproduction factor, was pretty important. So we know that in the territory, things such as wind cassia, the pronto sorghum, the Milgara butterfly pea, Cattenbora Rhodes grass, and the jumbo sorghum are going to be excellent resistant crops. What I did also find, though, is that cavalcade, which is that common hay crop up here and is also frequently used as a rotation, is highly susceptible. And we have sun hemp, which is another frequently used crop that is moderately susceptible. Another way to look at that is just through this simple table. And so this just kind of describes the different reproduction factors that I was finding for each one of these. Now, really, what does that all mean? I guess in summary, we have a lot of work to do on different management strategies that we can give to growers to combat root knot nematodes in the territory. What we do know is that throughout this project in the past year, we have boosted the molecular diagnostic capabilities here in the territory to be able to do our own work instead of sending it interstate. I've been able to go through 260 different samples and can report back that root knot nematodes are a very common problem that affect at least 30 different crop species. Meladogyne javanica is the most important species, covering 37% of all detections, followed by the guava root knot, which is Meladogyne enterolobii. And at the moment, because of some of those inconsistencies with different resistant crops, and because of the struggles that we may have with some of our former management strategies, such as turning off irrigation, we really need to focus on using hygiene and good solid biosecurity practices to prevent the spread of root knot nematodes as one of our paths forward. So this project will continue on for the next couple months, and it is in collaboration with many other state and territory jurisdictions. So I'm sure there's a few people listening that are also participating in this project. So thank you for all of the hard work that you've put in with the project in total. And I'd also like to thank all of the NT-DAF staff that have supported me, the QDPI staff, and all of the growers, community gardens, and households that have allowed me to sample on their properties. Thanks for your time.
Thank you very much, Mel, for that interesting talk and sharing your findings on guava root knot nematode in Northern Territory. We have 5-minute questions for Mel. So if anyone has a question from audience, please unmute and you can ask or put your questions in the chat box so we can discuss. So
can I ask a question? My name is Shamsul Buyan. I'm from Sugar Research Australia, Brisbane. So my question is, does it mean that we have the guava root knot nematode already present? So what is the implication to the other crop? Do you think it is now? What can we do in the next at this moment?
For the guava root knot nematode? I guess the good news that I found is that surprisingly, we haven't had a lot of spread across the territory. I thought there was going to be a lot more than there is, even though roughly 30% of the detections have been enterolobii. I have been really focusing on specific areas where I thought I was going to find it. And because I have noticed that it seems to be concentrated on certain things and it's following a lot of different people's practices. So most of the detections are actually based on residential properties. So that includes all of the suburbs around Darwin. And I've only found it on 6 commercial farms, which means we've been able to trace back how those commercial farms have gotten it as well. And it all seems to be related to the sharing of contaminated plant material. And so it doesn't seem to really be spreading outside of that. So there have been only direct links. I guess in the territory, I think that's why I really believe the messaging around keeping things clean is going to be the best way to prevent it. I think in terms of Queensland, the QDPI team would probably be able to share more on some of the strategies that they have for your crops over there. But in the territory, I think I'd be a lot more concerned about some of the other root knot nematodes that we already have here.
I guess, can I ask another question or just, that's fine if I...
We have another question in the chat box. So if we can go for that. So we have one question from Pragya from Agvik. And she's asking, so the table showed that you identified nematode in table grapes. Did you consider vine grapes? Do nematodes infect both or only table grapes?
Yeah, so I was only looking at crops within Northern Territory. Unfortunately, we're a little too hot here for wine production. There used to be one wine grape vineyard down in Alice Springs, but it no longer exists. So I looked at table grapes for that particular reason. I do know that wine grapes are susceptible. There's quite a few different types of rootstocks that are resistant to root knot nematodes, though, and I think those are frequently used across Victoria, New South Wales, and South Australia.
Yeah, thank you, Mel, for the clarification. Appreciate that.
No worries.
Okay, thank you very much, Mel. Now, in interest of time, we will move on, but please feel free to discuss any question in the chat box with Mel. Now, I would like to introduce our second speaker for today is Dr. Sonu Yadav, and she's going to present on the metabarcoding of soil fungal microbiome and its environmental drivers across temporal and spatial scales. Whenever you're ready, Sonu.
Thank you, Shreya. Am I audible or my slides are up there?
Yes.
Yeah. Hello, everyone. My name is Sonu. I am the principal molecular scientist here with the Department of Agriculture and Fisheries. And I would also like to acknowledge that I work and live on Larrakia Nation, and Larrakia people are the traditional custodians of this land. And I also pay my respects to elder past and present. I'm going to talk about a project we recently finished on metabarcoding of soil fungal microbiome. So I'm just going to give a bit of background and the methods we used and some of the findings and summarize. I also would like to thank Elnas, who worked on this project for the last 2 years and did lots of analysis. So yeah, with the background, Northern Australia is a big landmass, and it's also a high-risk zone for environment and plant biosecurity. Surveillance can be difficult because it's pretty large with sparsely populated regions. It could be pretty remote as well, and identifying and doing surveillance of pests and pathogens could be really labor-intensive. And time-consuming, and sometimes, as Mel mentioned in her presentation, where it could be very difficult morphologically. So application of molecular techniques and next-generation sequencing can, up to some extent, help and facilitate this early detection and faster detection of the pests and pathogens. And one such technique that's been, that's growing in the biosecurity sector, but in general as well, is the environmental DNA technique. So this slide just shows you many different applications of this technique, and it's been in use since some time now, and it was started with diet analysis, but now it has been expanded to a lot of different areas. But the one area that I'm going to talk about today is the community biodiversity assessment. Which is basically, you could use a lot of different sample types. So you could use soil, water, sediments, or even invertebrate-derived DNA. So insects to then assess the vertebrate biodiversity. And in terms of methodology, this is just a very simple schematic to show what happens. It can change according to studies. So first thing is sample collection, and it could be environmental DNA, and then extracting DNA from the sample, doing PCR. So many different target or barcodes could be used. Library prep, sequencing on high throughput machines, and then finally doing the bioinformatics and analyzing. So by combining this environmental DNA sampling and metabarcoding, we could produce large amount of data set, and they can tell us a little bit more about like what texts are there and their relative abundance and diversity. So with this project, we wanted to develop a metabarcoding protocol that were relevant to Northern Australia. And we used soil. So we assessed soil fungal biodiversity, and we did it across many different times and across a broad spatial gradient. I'll talk about it a little bit more in next slides. And then we also tried many different DNA extraction protocols and sequencing methods. And finally, we also looked how environment and climate and soil properties can also influence this microbiome composition. And by doing so, we enhanced our own capacity and capability to do such work in Northern Territory. So with methodology, this map here just shows you the different spots we collected samples from, and the points are on annual mean temperature. So the orange is lower temperature and green is higher temperature. And the shapes are different climate zones. So we covered savanna, arid, tropical, and equatorial zones. We collected samples from 30 sites across 4 time zones in 12 months. We covered 4 climate zones, we covered 4 vegetation types, 8 soil types. And as you can see, it's a pretty broad, wide landscape, and we couldn't do everything by ourselves. So we collaborated. So I think this was one of the really important part of this project is this collaboration. So we had police officers collecting soil samples for us, and biosecurity officers. We had Indigenous rangers collecting samples. So we just sent collection tips and just a whole kit and guidelines on how to collect. And then they were just sending us samples 4 times a year. So it was really exciting and interesting part of this project. So once we had samples collected, we applied kit method, which is commercially available soil kit extraction methods. And then we applied non-kit method, which was developed in 2012, and it uses a large, more proportion of soil, and it skips the lysis step in the extraction. We targeted ITS region because it is considered as a universal barcode. There's a more database available for it on public database, so it kind of helps to map reads. And then we tried 2 clustering approaches. So basically, when the sequences are produced, how they are clustered, so we can use that genetic data. So we've used amplicon sequence variants, which keeps more variants and different after filtering out bad reads and error corrections. It keeps variants. And then we also tried operational taxonomic units, which is more traditionally used in these studies. And then we also did variable extraction. So we extracted environmental factors like vegetation type, soil type, climate. We also recorded collection time. So a lot of these things were extracted from public database. Then we did bioclimatic variables as well. So precipitation and temperature data downloaded from WorldClim. And we also measured soil pH, nitrogen, phosphorus, and potash level in lab. And then finally, when we had all our data, we did alpha and beta diversity analysis. And then we also did redundancy analysis, so RDA analysis, to look at the impact of different environmental factors on composition. And then we also did some relative abundance studies, so which is the most abundant group and how does it changes with time, climate, and vegetation. So in the interest of time, I'm not going to talk about all of that. I'm just going to talk about the temporal effect, and a lot of this analysis was done in our studio. So key findings. I'll start with diversity. So this graph here, it shows the ASV here and OTU, and the dots are color-coded based on the kit and non-kit method. So the green is non-kit method and the purple or blue is kit method. There was no significant difference in the ASV and OTU data with richness. So they almost had kind of similar means. But there was a bit of intra-sample spread and variability in kit method compared to non-kit method. The other measure we used to calculate alpha diversity was Shannon index. So it calculates richness and evenness. And in this case, this is ASV and this is OTU data. And in this case, we did have a significantly higher Shannon index for OTU for ASV dataset compared to OTU. And then we also, within also ASV, we had a more larger spread for non-kit method compared to kit method. So there was some difference observed based on the methods that we used. With beta diversity, so this graph is ASV and this graph is OTU. And we did observe significant difference between ASV and OTU. So the composition and the diversity here in ASV was similar for both the methods. So for kit and non-kit method, it's sort of similar composition. So there was no significant difference within ASV. But for OTU dataset, there was a significant difference between non-kit method and kit method. So the non-kit method I did or had a different composition structure compared to kit method. So then we did some analysis over time. So whether we find diversity changes with time. This first graph here shows you the total Shannon index across all methods, and then we laid it out across the climate zones. So the tropical climate zone had significantly higher fungal diversity compared to all other 3 climate zones. And then when we looked at different ASV and OTU dataset, in general, ASV had higher diversity across for all the climate zones, so equatorial, tropical, savanna, and arid. And the diversity was also highest at time 3, which was October and November. So it's sort of like the wet season here. And so it's kind of high in all of these except in arid zones. So arid savanna, in arid time 2 and time 3, they had a sort of similar diversity index here. So, but these differences were not significant, statistically significant. It was just a bit higher at time 3. So this is a RDA plot just showing composition, and this is for the first 2 plots are above to kit method and the below to our non-kit method. So kit method actually had like these 2 separate clouds. So tropical and equatorial form one cloud, and savanna and arid, they had a sort of different cloud, which was lost in non-kit method. And then temperature and precipitation came out as the most important variables that were influencing the composition. In terms of relative abundance, for kit method, we found aspergillus is the most abundant one, which was not true for non-kit method. So non-kit method had Melacesiaceae as the highly abundant, and which was only detected by non-kit method. So it was not detected by kit method. ASV and OTU did not differ significantly in overall relative abundance. It was more about the method of extraction. And the highest number of reads were just unclassified. When we looked at the temporal change across different time zones, so the first 2 graph, this A and B are kit, and then C and D are non-kit. So for aspergillus, which was the highest one in kit method, there was a significant change of relative abundance across time, and it was highest at time 4, which is Feb and April. So the basically the tail of wet season and almost the start of dry season, which is wet season is more like hot and humid, and dry when the temperature starts cooling down. And in non-kit method, Melacesiaceae also showed a similar trend, so increasing relative abundance at time 3 and 4, so October to April. So in summary, we did find variation across extraction methods and the sequencing grouping methods, which suggests that increasing more and more methods and multiple approaches could improve our fungal diversity and composition assessment. Especially like ASV was good at finding diversity and OTU was good at finding unique families. Precipitation and temperature were the common drivers. They were the key drivers in variation, fungal composition in both the methods. And there was a change in the relative abundance across time. There was also change across vegetation type. I'm not presenting that today, but we have a paper that's published. So if you're interested, you could also go and check it out. But yeah, as I mentioned, the engagement part of this work was really something very new, and it was very good. So, you know, we were talking about the environmental DNA and new techniques of doing biosecurity, and it established that relationship between us doing this, you know, lab work and doing the next generation work, and then also translating it to a wider public. And it also increased the capacity of us doing all this work in-house. So in terms of benefits and limitations for this method, eDNA metabarcoding could help, especially when there's complex environmental samples that PCR products are mixed, and it also helps in broader biodiversity monitoring, and also to include to make it as a community science project. But it does have limitations in methodology, so DNA quality or PCR and primary specificity. There is still incomplete reference database, and then also the read counts. Sometimes it's possible to miss low abundance taxa, and then the biologist or the scientist also need to have that expertise to validate the data and the accuracy of the data. With that, I would like to acknowledge a lot of people who were involved in this project in one way or other. My team, and then the police officers and biosecurity officers, biosecurity team, plant biosecurity team. A lot of people did, sampling or helped us in one way or other. This is the article that's been published now. So if you want to read more, there's so much more that I couldn't present, but yeah, thank you very much, and happy to take any questions.
Thank you very much, Sonu, for a great presentation and sharing your insightful work. We are open for the questions now. So if anyone has a question, please unmute and ask question.
Can I ask a question?
Yeah.
So this is a great presentation. I guess the one thing I would like to know, because of this community kind of study, how it possible actually, can that be actually as a practical application in a soil health study or the regeneration study and this kind of thing using the fungal community analysis?
Yeah, I think it depends, if you're targeting, it depends if you're targeting a particular species. So the assays we develop or the methodology changes. So it could be used for soil health or for detecting a particular pest or pathogen in the soil. But yes, how we develop and how we design the experiment is different on what our questions are. Sometimes for a particular target species, we might have to do a bit more work on the specificity and the sensitivity of the assays. But there are always those opportunities to improve the technology and then improve the protocols, especially the lab protocols. But it could have that application of applying it into soil health, or even I think, just communicating and connecting with the wider public, and then saying, this is what we do, and just kind of, also helping people to understand what we all do as well. So yeah, I think it has a lot of benefits.
Okay, thank you, Sonu.
Is there any question? Okay, no more questions. Thank you, Sonu. So that brings us to the end of today's seminar. And I want to extend a massive thank you to both our speakers, Mel and Sonu, for sharing their expertise, and to all of you for your active participation and attending this seminar.